Welcome. I'm very happy that so many of you are here after the conference dinner. This is often not the best time to give a talk, but it seems that you all got up to attend the talk. Thank you very much for this in advance. So my talk is going to be about statistical significance in biomarker discovery. And let me first describe why statistical significance is such an important issue in biomarker discovery. If you listen to the talks at this year's summer school and at the previous editions of our summer school, then a reoccurring theme is that complex diseases, complex traits are not determined by single genes, but rather by large groups of genes. They are polygenic in architecture, in their genetic architecture. This has a lot of implications for the data mining, for the machine learning efforts that we perform here. In particular, there is the need in feature selection, in genome-wide association studies, to look for combinations of, you could call it molecular factors, SNPs, genetic variants, genetic loci, so combinations of these that together are associated with the phenotype. Now, you may remember that in Tübingen, I gave a longer course on data mining in the life sciences, and I talked a lot about the computational issues, the computational challenges that we face in this combinatorial feature selection problem. How difficult it is to search this huge space of candidates of combinations of molecular factors. And you may also remember that I mentioned at the time that there are two problems. There are not only these computational problems, there are also the statistical problems involved in searching such a large candidate space. But I said in many talks, today we are going to focus on the computational problem, and we are leaving the statistical problem aside. So we turn this feature selection problem into a pure feature ranking problem. We just want to find the top scoring features. Today, I'm turning this argument upside down. Today, I will focus on the statistical aspects, which are so often neglected and ignored when we do data mining, when we do feature selection in high-dimensional spaces. What is this statistical problem? The statistical problem in feature selection from high-dimensional spaces is that by enumerating all these candidates and testing them for association with the phenotype, you are testing one hypothesis for each of these candidates, which means if you are considering millions and billions of candidate patterns, candidate combinations of SNPs or molecular factors, then you are testing millions or billions of hypotheses. And even with a beginner's knowledge in statistics, you'll be aware that in such a scenario, you have to correct for the fact that you are performing multiple hypothesis testing, although data mining largely ignores this problem. Today, I want to change that and look at that problem of multiple hypothesis testing in feature selection in data mining. More specifically, from a data mining perspective, you can think of the problems that we are studying here in genome-wide association studies with higher order combinations of SNPs as a so-called pattern mining problem. Now here's a toy example that describes the typical setting in a pattern mining problem, in a so-called discriminative pattern mining problem. We are given two classes of patients. Let's say that the class 1 is the class of individuals that do not respond to a drug. Class 2 are the individuals that recover by this drug treatment. And we have binary features. That's the underlying assumption that I make here and also for the rest of the talk. We assume we have binary features that describe properties in our applications of molecular properties of these patients. So each row is one feature. Now the problem of feature selection is to find the features, and in particular the single features, that correlate most with the class label, with the phenotype in our words. In pattern mining, we are looking for higher order combinations of these features. So we are combining several of these features, for instance, by taking their product, to then see whether this higher order combination of features is correlated with the phenotype. That's a typical data mining problem to look for, a typical pattern mining problem to look for higher order combinations of binary features. What would be a typical statistical test that we apply in this setting? The most famous one is Fisher's exact test, based on a 2x2 contingency table, in which Y is the class label. Y equals 1 means it's a member of the first class, Y equals 2 of the second class. S1 or S is such a pattern. Think of S as the product of a number of features. Here from this example, let's say the product of the first 2 rows. So if that product is 1, then we increase the counters in this first column here, and if it is 0, we increase the counters in this second column. Very important here are the following variables. A is the number of times the pattern S occurs in the first class, X minus A, the number of times it occurs in the second class, and the sum of this, the total number of occurrences of that pattern, is X. The size of the first class is N1, and the size of the total data set is N. And typically, we compute p-values based on such a 2x2 contingency table using Fisher's exact test by the so-called hypergeometric distribution, which assumes fixed total marginals, so the total number of occurrences fixed, the size of class 1, and the total size of the data set. Now, let's state a few key facts here. Some are obvious, but let's still summarize them here. Each S, each pattern, and each contingency table that we test corresponds to one hypothesis that we are testing. To control the family-wise error rate, so the probability of detecting at least one false positive, one pattern that we incorrectly assume to be associated with the phenotype, we have to perform multiple testing correction. And there's a rich literature in the statistics community about how to perform multiple testing. The classic approach here is the Bonferroni correction, which is still widely used in statistical genetics, and we'll focus on this here. So the Bonferroni correction means you correct the significance level by dividing it, alpha, by dividing it by the number of tests performed, m. And thereby you can make a statement about the family-wise error rate of the entire set of tests that you perform. If we didn't do that, if we just ignored the problem, then we would run the risk that in our very high-dimensional spaces in genetics, we may discover millions and billions of false positive associations. Now, what can we do about this problem? This problem seems so overwhelming, and in fact so overwhelming that in data mining for decades it was an accepted fact that we just have to ignore this problem because we cannot solve it in our extremely high-dimensional spaces. But recently, there was new interest in this question, and suddenly we start to reconsider this issue and to investigate whether it might be possible, at least in some classes of problems, to correct for multiple testing, even in data mining in very high-dimensional spaces. And key to this new development is a rather old paper, 25 years old, by Terone from 1990. And Terone made the following observation. For discrete test statistics, so the test statistics are computed on discrete data, such as in Fisher's exact test, there's a minimum obtainable p-value, p-min, which is, to put it a bit informal, which is saying even in the optimal configuration, in the optimal configuration of this contingency table, we'll obtain a minimum p-value, which is larger than 0. This is a very important observation here. So if the counts are perfect in the sense that the pattern always co-occurs with one of the two classes, then we'll have a very significant p-value, but that p-value will be larger than 0. It's the so-called minimum p-value that we obtain for this discrete test statistics. Now, the second important insight is this minimum p-value that we obtain may still be larger than the corrected significance level corrected by Bonferroni correction. So the minimum p-value is larger than the corrected significance level. And Theron gave a name to those hypotheses for which the minimum p-value is larger than the corrected significance level. It's the set of untestable hypotheses. These hypotheses can never become significant. This is Theron's key insight. And based on this insight, he proposes the strategy to ignore these untestable hypotheses when counting the number of tests for Bonferroni correction. And he also shows in his paper that if the p-values of the tests are conditioned on the total marginals of the contingency table, as they are in Fisher's exact tests, then removing the non-testable hypotheses does not change the family-wise error rate. So in short, it's a way to get a much better, much lower correction factor. The number of tests here that we correct for is called the correction factor, to get a much better correction factor in Bonferroni correction. But there is a big difficulty in this, a big computational difficulty, because there's an interdependence between the number of tests that we correct for and the number of testable or non-testable hypotheses. The number of testable hypotheses depends on the significance threshold, which depends on the number of hypotheses that we correct for. The number of hypotheses we have to correct for, however, depends on the number of testable hypotheses. So there's an interdependence between these two terms. To put it more abstractly, if k is the number of tests that we correct for, and m of k is the number of testable hypotheses, so the number of hypotheses that we have to correct for at level alpha divided by k, then we are looking for the smallest possible correction factor, such that this correction factor is at least as large as the number of testable hypotheses. So it's an iterative, so you can solve this problem iteratively, as we'll see on the next slide. We have to find, or you could rewrite this here as the difference between k and m of k, and we have to find out when this difference is 0. So when is k larger than m of k? Now Theron proposed the following algorithm here, and as this is a machine learning network, I decided also to include some pseudocode today in case you're interested in implementing these approaches yourself. This one is rather easy, looks rather easy at least. So Theron's procedure is that we set the correction factor to 1. And then we increase it by 1, check whether k is still smaller than m of k, whether the correction factor is still less than the number of testable hypotheses for this particular k. And if it is still smaller, then we increase k again. So step by step, we increase the correction factor until it exceeds the corresponding number of testable hypotheses. That's Theron's approach. Of course, I haven't given any details about the m of k. How expensive is it to evaluate m of k? Assume for now that we just compute all p-values and check which of the all minimum p-values for all candidate patterns, and then we check which of these has a minimum p-value below this significance level alpha divided by k. So this was the paper from 1990. And it was almost forgotten for 23 years and had hardly any citations. And then in 2013, two Japanese groups, Yun Sese's lab and Koji Tsuda's lab, whom some of you might know, together published a paper in PNAS about discovering gene regulatory networks, in which they drew a connection between Theron's approach and what is called frequent item set mining in data mining. So for those of you who don't know what frequent item set mining is, if you are given a set of transactions, so a set of sets, you could also call it. For instance, the shopping lists of a lot of supermarket customers. And you are then looking for subsets of elements that very often co-occur in this database. So for instance, products that are often co-bought by the same customer. They are frequently co-bought by the same customers. Then this would be a frequent item set, this subset of products that the customers always buy together. And there are tons of algorithms for this problem and variants of this problem in data mining, finding subsets of objects that occur more than some frequency threshold, more often than some frequency threshold in a large database of sets. And now what Terada and Tsuda and Sese pointed out in 2013 was that if we assume that the frequency of the pattern is upper bounded by n1, so the size of the first class, and lower bounded by 0, of course, then the minimum p-value that can be obtained for this pattern, S, decreases monotonically with its frequency. And the consequence, the big consequence of that is that one can use frequent item set mining to find all S that are testable at level alpha. So there's a connection between being frequent and being testable. That's the insight from Sese and Tsuda. And that helps us greatly to compute the M of k that I talked about before, like the number of testable hypotheses for a given correction factor. Now, it's well known empirically, and also it makes theoretically sense from data mining, that it's very expensive to run frequent item set mining with a very low frequency threshold. For instance, if we used this scheme here, then the first iteration would take forever. As a side remark, it would also give us the entire solution, if you think about it, and we wouldn't need iterations anymore. So therefore, Terada and Tsuda and Sese proposed another search strategy, which is the so-called decremental search. So they set k not to be 1, but to a very large value. You can define this precisely, but this is just a minor detail here. So while the correction factor is larger than M of k, the number of testable hypotheses, we reduce k now. And we compute M of k by frequent item set mining on the data set D with a frequency threshold that corresponds to being able to reach a minimum p-value of alpha divided by k. And this we express by this function psi. Psi inverse is giving us the corresponding frequency threshold for alpha divided by k. So instead of going up with k, we go down with k, and as soon as k is less or equal to M of k, we stop this loop here, and then we return k plus 1. This is then this minimum k that we are looking for, which is larger than this corresponding M of k. Now, what's the practical effect of that? Is that just a theoretical exercise which has no practical impact? I would like to show you one example, one counter-example to be precise, which shows you how interesting and how important this is. So this is from a graph classification benchmark dataset on which you can perform pattern mining. And if you just compute if the number of all patterns in this data set. This is shown here on the y-axis. And here on the x-axis, we show the size of the set of patterns that you're considering. So if you compute the raw number of patterns in this data set, then we have a drastic increase in the number of patterns that we have to correct for, the larger these patterns get. So hundreds of millions at least, if not billions of patterns have to be tested, and we have to correct for all of these tests. However, if you use Tarone's trick and exclude the non-testable hypotheses, this number of tests that you have to correct for reduces to roughly 10,000, even when you have no constraint on the size of the patterns. So the correction factor gets several orders of magnitude smaller by excluding the non-testable patterns. And this has an immediate effect on our results. So on this very rather old and very well-studied data set, one could only detect one significant subgraph so far if we upper bounded the size of the candidate set by 6 it is. And now with this improved Tarone scheme, we can suddenly discover between 2 and 4 significant subgraphs. We can even discover significant subgraphs when we do not upper bound the size of the set of features that we are considering, so the size of the pattern. And I found this very impressive. And I remembered conversations with Koji Tsuda in Tübingen, in which we said, in graph mining, which is one branch of pattern mining, correcting for multiple testing is absolutely hopeless. And you cannot make any significant statement here. But these results contradict that statement that we agreed upon like 5 years ago. It is possible, in particular on the data sets that we typically study in graph mining. So let's talk a bit more about graph mining, this one instance of pattern mining that is a very difficult sub-branch of pattern mining, because it's even more computationally expensive than the other branches of pattern mining. So what is significant subgraph mining? Well, in this problem, each object is not a set, but a graph. So you could say the elements of the set are now the nodes in the graph, but on top of being in the same set, we also have a graph structure that connects these elements to each other. And again, we have two groups of objects, class 1 and class 2. And that's a very famous setting from chemoinformatics, in which we have two sets of molecules. The first set has a particular function. The second set does not have that function. And now we want to find out in frequent subgraph mining whether there are subgraphs that are overrepresented in one set, in one class, but not in the other class. And for instance, here this subgraph, this red subgraph here, AAB, occurs in class 1, but not in class 2. This is and it occurs 3 times. So this is the type of significant subgraph that we are looking for here. But as I said, for a long time we thought this is hopeless to solve. It's hopeless to try to find a solution to this problem, because of course the number of tests here is exponential in the number of nodes in these graphs, if we consider all possible subgraphs for enrichment in this data set. Now, when we read that paper by Tsuda and Sese in the reading group of my lab, we wondered whether this decremental search strategy that I just described, like setting K to a very high level and then lowering it step by step, is actually practical in graph mining. Because it means that you have to perform a lot of frequent subgraph mining iterations for different values of K. And it turned out that in most datasets, the final K, the stopping point, the K that you return, the solution, is quite low. So it means you have to perform a lot of iterations of frequent item set mining for different values of k, starting at a very high value of k, going down to this very low optimal value of k. So that seemed impractical. And therefore, we thought about an alternative solution, an alternative search strategy that would allow us to find this optimal k faster. And we wondered whether it's always so expensive or whether it's always practically infeasible to do the incremental search of Tyrone. And the naive answer is yes, you should not run frequent item set mining on a graph data set with a frequency threshold of 1, because this means you have to enumerate all graphs in the graph database. And you know how many isomorphism checks and subgraph isomorphism checks are involved in this, so this will take forever. So a naive implementation of Tyrone's algorithm will not give the solution. But there is a property of frequent item set mining which we can exploit. The property is that almost all, if not all, frequent item set mining algorithms are branch and bound algorithms that enumerate the frequent patterns one by one. So they explore the candidate patterns in a tree-structured space, and they walk through this tree and return one pattern after the other. So we always have a counter that increases. And now the following insight is important. If we do the incremental search in which we increase the k one by one, then we can stop the current iteration if the counter for the frequent item set, so I call this counter here fs, if that counter for the frequent items exceeds the current correction factor. That's what this complicated term here describes. So if the number of frequent items, as soon as the number of frequent item sets exceeds the correction factor that we are currently using, we know that this correction factor is too low. And therefore, we can increase the correction factor and proceed to the next iteration. So this is what we call an incremental search, like in Tyrone's paper, but with early stopping. So we can stop these iterations as soon as the number of frequent item sets exceeds the correction factor. And now I show you the runtime results that we obtained on the same data set as before using these different strategies. The two interesting results here are shown in blue and in green. So green is the decremental strategy by Terada et al. Green is our strategy, and on this data set it's one order of magnitude faster, actually, to go the seemingly expensive way up from k equals 1 rather than down. So one order of magnitude on one data set. Is that so impressive? Well, let's look at more data sets, and let's look at the correction factor across the data sets. So first of all, what we could confirm on a large number of data sets is that this trick by Tyrone really makes a difference. The red line here is the naive count of patterns. The blue line and the green line are two variants of Tyrone's approach. So you can see here that on several data sets there's a dramatic difference between the red and the blue line, like here and here and here. And on some other data sets the difference is smaller between the correction factors. And I can already say this now, understanding this in detail, why some data sets can be lead to a dramatic reduction in the number of tests that we have to correct for and others don't, this is still an ongoing research topic for us. So empirically, we observe everything from a small improvement to a huge improvement. That's the short story here. Now the runtime, what is interesting here is among all of these different variants of Tyrone and our approach that we implemented, the interesting thing is that the light blue line here, this is Terada's search strategy is always orders of magnitude slower than the incremental search and its variants here. So these are the blue and the green lines here. So there's, across all data sets, an improvement by using this incremental search with early stopping. So far, we have just improved the computation time of how to find the optimal correction factor. However, in this work by Tyrone and by Terada and by us until 2014, we did not take one important point into account. Of course, in pattern mining, where we combine single features into larger groups, there are very often sub and super pattern relationships between these patterns. So patterns are correlated with each other. Patterns constitute dependent hypotheses. There's dependence between these tests that we conduct. And we did not consider or use that dependence so far. Informally, one could say the raw count of the number of patterns that we are testing is much larger than the underlying real number of hypotheses that we would have to count if we take all of these dependencies into account. Now, what would be an approach to take these dependencies into account? The most famous one are the Westphal-Young permutations from 1993, in which the class labels are repeatedly permuted to approximate the null distribution. And this is one strategy how to take this dependence between tests into account. However, how do you efficiently perform these thousands of permutations on millions or billions of patterns? That's a huge computational problem. And there was one approach from the Sese lab which addressed this problem, but it either suffered from memory or from runtime problems. So we saw a lot of room for improvement here and saw the need to develop an algorithm that is both fast and memory efficient in performing these Westphal-Young permutations. Now, how does this algorithm work? Here's again the pseudocode which gives you a number of tricks that we use here. So what's the input to our algorithm? Again, we give the algorithm a data set, D, of transactions. So these are these supermarket lists that I talked about, or the graphs, the molecules, or they could be electronic health records of patients. Then we have class labels Y, so whether someone is a case or a control in a clinical setting. We have a target family-wise error rate alpha, and we have the number of permutations J. And what we would like to get in the end is the corrected significance threshold delta star. And what we do here now is that we do again an incremental search, and we start by setting the minimum p-value across all permutations to the maximum possible value, to 1. And then we start searching the space of patterns. I mentioned before the typical way how you search the space of patterns is in a tree-like structure. So we do a depth-first search on this tree and look at one pattern at a time. And we compute the p-value of the minimum p-value of that pattern, or the p-value of that pattern across all the permutations. This is a big difference to the previous approach by Terada et al. They started frequent item set mining anew in each permutation. We do all permutations on one pattern at once, and we see whether we have to update the minimum p-value for a subset of the permutations. And as I mentioned before, there's a direct relationship between the minimum p-value a pattern can achieve and the minimum frequency threshold that you should use for finding these patterns by frequent item set mining. So we can constantly, in this process, increase this frequency threshold. And thereby, we can solve this problem with one single iteration of frequent item set mining. One single iteration of frequent item set mining that changes the frequency threshold along the way, but it's still called only once. And this makes this approach very fast. And as you may have noticed, we don't have large memory needs, so we don't store a lot of information, so it's also memory efficient. So here's a summary of what we do in this algorithm. We follow an incremental search strategy, so we start again at a very low correction factor, rather than the decremental search strategy of the algorithm fast Westphal-Young by Terada et al. We perform one single iteration of frequent item set mining instead of one iteration per permutation. We do not store the occurrence list of patterns, so in one variant of the algorithm they have to store in which transaction does which pattern occur, which is a huge matrix in the end. We do not need to store that matrix. I mentioned that Westphal-Young computes the null distribution of minimum p-values across all the permutations. We do not have to store, what's that? Okay. We do not have to store the upper I minus alpha quantile of this distribution. This leads to a huge extra runtime effort in Terada's approach, that they have to store the entire null distribution. We only consider the lower alpha quantile of that distribution. And because of that, we also can avoid some expensive iterations of frequent item set mining, because finding the upper quantile of that distribution of minimum p-values corresponds to performing frequent item set mining with very low frequency thresholds, and that's very expensive. So we get rid of a very expensive part of the computations. And we share the p-value computations across all permutations. Now, what's the impact of that? We test this again across a large number of pattern mining benchmark data sets. In the execution time, you can see here that we win at least 1 order of magnitude compared to the other approach. And this is a very well studied domain, in fact. So gaining 1 order of magnitude is a big thing is a big thing here. And you can see that the point that I mentioned before, that we don't have to run frequent item set mining with very low thresholds. You can see this in this plot here compared to the algorithm by Terada et al. So the minimum frequency threshold that we use in this data set here, for example, is 10, and in Terada it is 4. And there's a huge difference between running frequent item set mining with a frequency threshold of 4 or 10, because you can prune away a large part of the search space if your threshold is a bit higher. So, and you see this constantly across all the data sets, that our minimum frequency threshold is much larger than that of the other algorithm. And here you can see the peak memory usage. In green, the usage by fast Westphal-Young by Terada et al. In blue, our memory usage. And in red, the memory usage by the frequent item set mining algorithm. And you can see that the blue and the red bar are almost the same height, so we are quite memory optimized here. So this is like the minimum we need, the memory requirement of the frequent item set miner, and we don't need that much extra memory, whereas fast Westphal-Young sometimes peaks very much. So due to these memory requirements, we can now run significant pattern mining on data sets which could not be analyzed before due to large memory requirements. And this plot here illustrates that correcting for the dependence between tests is really necessary and important. So the empirical family-wise error rate that we get in this approach that I just described, based on the Westphal-Young permutations, is indeed close to the target significance level of 0.05, whereas using the Terone approaches and our approaches from the first part of the talk gives a family-wise error rate around 0.02. So we need to take the dependence between tests into account. And now to the final part of my talk, which is an application of these ideas to association mapping in genetics. It's concerned with the discovery of genetic heterogeneity in the genome. The classic meaning of genetic heterogeneity is that the same phenotype may be caused by a set of different genes. This is the classic meaning in medical genetics. I think nowadays it often also just refers to the fact that different individuals differ in some part of the genome. But I mean the old definition of the term. So one phenotype is caused by a number of different genes. Now, if we have this situation that a number of different genes cause the same phenotype, then the correlations between sequence variants in these single genes and the phenotype may be too weak to be detected, whereas considering the whole group of genes together under a model of genetic heterogeneity may give a correlation that is strong. This is one hypothesis, but of course the obvious question to answer is how do you find this group of genes that is jointly so strongly correlated with the phenotype? Well, the only current way so far was to consider fixed groups of variants, for instance, all variants within a gene together, or to have some other criterion how you define the set of genes or the set of SNPs that you consider for jointly exhibiting genetic heterogeneity. So you always had to define the starting point and the end point of intervals, for example, if you looked for intervals, contiguous subsequences of the genome which jointly underlie genetic heterogeneity. Now the contribution of our algorithm, which we call fast automatic interval search, is that it finds all contiguous sets of variants that are significantly associated with a given phenotype under a model of genetic heterogeneity. Now what does this model look like? Let me illustrate the problem. So we are given here in a toy example 6 plants with different colors. We call the two groups cases and controls. And these here are bit sequences representing their SNPs. Now rather than just looking for a single SNP whose state is strongly correlated with the phenotype, we look for intervals such that members of one phenotypic class tend to have at least one minor allele within that interval. And it's right, I here assume binary SNPs. So I assume, for instance, that we work with Arabidopsis thaliana, where this is really the case in the lab strains. So we have binary SNPs. One represents the minor allele, and our idea is having at least one minor allele in a given interval should be correlated with the phenotype. And this random variable here on the right describes just that, whether a minor allele occurs in that interval or not. However, unlike all previous approaches, we do not fix the number of intervals that we look for. We would like to consider all potential intervals in the genome. So we don't want to restrict the search space by hand. We want to search all possible intervals, which directly may lead to a huge multiple testing problem, because then we have order length of the genome squared tests that we have to correct for. So both. We would like to have both proper correction for multiple testing and the ability to find any interesting interval in the genome. How can we achieve both here? Well, we achieve both by using Theron's approach in our algorithms that are further developments of Theron's approach, based on a few insights or properties of this particular application which we can exploit to further speed up the computation. Now, one insight or one important fact is that the representation of a subsequence of the genome that I talked about is mathematically the maximum over all the SNPs in that sequence. So when I say if there's at least one 1 in that interval, we represent it by 1, and if there's no minor allele, we represent it by 0. That is identical to saying we take the maximum over the values of the SNPs in each interval. Now, another fact that follows from that is the longer you make the sequence, the more likely it is that the sequence at some point will contain a 1, a minor allele, and therefore will be represented by 1. And now how can we use this fact? I talked a lot about patterns being non-testable because they are too rare, because they do not pass a certain frequency threshold. Of course, this phenomenon also exists in the other direction. If a pattern occurs in all objects, then it's not significant either, because it's represented in both classes, or it's very frequent in both classes. So we worked out the following bound. So in the original Terada work, they only considered the case in which we can prune away patterns that are very rare. And in this plot here, we showed that also for very frequent patterns, we can prune them away because their minimum p-value is actually very, very high. So Terada just lower bounded the minimum p-value for the higher frequency patterns here by a constant, and we showed that this minimum p-value actually in log space has such a W shape. So when the pattern gets very frequent, it again becomes non-testable. So insight 1 and pruning criterion 1 is if an interval is represented by 1 for too many individuals, then the interval is not testable. If a pattern is too frequent, then the corresponding interval is not testable. Pruning criterion 2 is also based on the maximum and on the fact that we use binary representation here. Once an interval is 1, we cannot make it 0 again by extending it because we are taking the maximum over the interval. That means if one interval is not testable because because the frequency of 1s in that interval is too high, then none of its super intervals is testable. To show the practical impact of what we are doing here, in this plot we show the length of the sequences of the genome sequence that we are considering in a simulation, and here the time taken to find the significant intervals, and the brute force approaches here are the two upper lines. One is using Westphal-Young permutations, the other isn't, and down here are the two variants of our approach of the fast automatic interval search, where the upper one is using Westphal-Young permutations, so considering dependence. The lower one is not considering dependence. And you can see here that there's a drastic runtime difference if we are looking at sequences of length 100,000. In simulations where we simulated significant intervals, also our approaches had a higher power than the brute force approach, and then univariate approaches that just look at one SNP at a time and taking dependence into account by performing Westphal-Young permutations also led to a slight increase in the power. One question that we asked ourselves and that also the reviewer wanted to know is, if you now compare this interval search to the standard univariate mapping approaches that you would use, like a univariate Fisher's exact test or a linear mixed model based using a single SNP. do you really find new regions in the genome compared to these univariate approaches, or do we just find intervals around a significant hit, and we just add some noise and some sequencing errors around a known hit? And we investigated this on GWAS datasets from Arabidopsis thaliana for 21 binary phenotypes, and we found out that 30% of the intervals that we find are located near a hit that we would find with a univariate method, but 70% of the intervals that we find are genuinely novel. So it's not that we just add noise or sequencing errors to the right and left of a known univariate hit. This leads me to the end of my talk. I hope I have convinced you that multiple testing in data mining and pattern mining is an important issue, that it is not as hopeless as we thought for a long time, and that a lot of very interesting questions remain to be explored. I mention a few here. The one point that I also talked about during the talk, we need a better empirical understanding of the impact of considering testability. Why is it improving the correction factor so much on some data sets and only a little bit on others? We still have in significant pattern mining a problem that also occurs in general in frequent pattern mining. Sometimes we get a lot of patterns in our solutions, so as we get many frequent patterns in frequent pattern mining, we sometimes get a lot of significant patterns in significant pattern mining, and we somehow have to summarize these patterns in order to make them interpretable for a human user of the software. Then, of course, in these genetics applications, it's very important to correct for confounders. And we recently worked on a test, on the extension of the Cochrane-Mentel-Haenszel test, which can correct for discrete confounders and still make use of this idea of the testability of patterns. Now, the future work on this idea will be funded by a starting grant, and I would like to mention that I have 2 postdocs and 1 PhD student position available in this project, if you would like to join this endeavor. So therewith, I would like to thank all my group members. I would like to mention that Felipe, who's the ITN student, it's one of the 2 ITN students in my lab. He was the first author on the majority of papers that I showed here. So a big thanks to Felipe and all the co-authors who worked on this project. Also to Mahito Sugiyama, former postdoc of mine who's now in Osaka, who also co-authored all of these studies. So it was a great pleasure working with them. The code is available from the homepage of my lab. So if you go here to BSSE MLCB, Machine Learning for Computational Biology, there's a link to research, machine learning and bioinformatics, and there's the code for all the algorithms that I talked about today. This is the new building we'll move into in 2020. If you join the lab, you might see the day. And these are all the references that I talked about, including the last ones, which are our work in that domain. Okay, thank you very much for your attention.